So today we continue on from chapter 10. Now you remember in chapter 10, the title of the chapter is cell division, mitosis, right? That's what we talked about in chapter 10. And it was very simple. Remember, the only thing that we had with mitosis was we went through a process by which we copied the chromosome if you were a prokaryote, or we copied the chromosomes if there was more than one, and that took a little while. They had to be synthesized, of course, if you're going to copy, and then they were going to be condensed and all that I showed you, and then there was prophase and metaphase. But in the end, what we ended up with at the very end was two cells that were, other than those rare little instances that I mentioned to you, they were identical to one another. They were clones. The purpose of mitosis is just to do that. It's just to make cells. It's all about quantity when it comes to mitosis. In my Bio 1 class, or Bio 2 class, we talk an awful lot about organisms and different reproductive cycles. The one we're most familiar with is ours, of course, but there are different ways of actually organisms that reproduce. And there are times during that process when they'll go through meiosis and mitosis. And I always tell my students, when you see mitosis, think quantity. All we're doing is making copies of cells. But when you think meiosis, and meiosis and sexual reproduction have to go together. You can't have one without the other. So remember back in the prokaryotic cell, the bacteria, it was undergoing mitosis and it was also reproducing. I mean, come on, if you're only a single cell and you split in half, then you've reproduced and you've also undergone cell division. It's the same thing with single cell protozoans, even though, they're not prokaryotes. But what happens in chapter 11 during this process that sounds like mitosis, which is actually meiosis, is this is where we get the differences that we see among organisms out there, the variability. This is something that if you talk to my Bio 2 students about this time of the semester, they said, oh my gosh, he says it all the time. I hammer on variability. That's because in Bio 2, the basis of that course is really how we're describing the diversity of organisms on Earth, but really more, and I won't say more importantly, but what's integral to that is understand how that diversity came about. So actually, I'm kind of starting in that variation stuff. I'm kind of starting talking about variation in chapter 11, this whole idea of sexual reproduction. If you have siblings from the same parents, they're not exactly like you. I mean, you might have an identical twin, of course, and all that sort of stuff, but you're all a little bit different. You might, some might have some characteristics more like your dad, or some characters more like your mom, or maybe a mixture of those. And variability is important, isn't it? Genetic variability is extremely important among a group of individuals, right? So everybody of the same species, something we call conspecifics. All us humans in the same room, we'd all be conspecifics. Why is variability so important, by the way? Why is it important that everybody in a room, or every roach behind a wall, or however you want to say it, why is it that they're all different? Yeah, that's right? Difference, competition. That means that some organisms are going to compete better than others, right? So there's going to be a struggle that occurs, right? And that struggle means you can only have a struggle if there's variability, right? I mean, two completely evenly matched football teams, right, are going to basically just keep each other in the same part of the field. That's where we get those, 0-0 or 3-3 games sometimes, right? Very evenly matched. So if everybody in the population is exactly the same, right, and something comes along and kills some members of that population, and the immune system can't fight it off, and everybody's got the same immune system, guess what? The species goes extinct. So that's the basis behind this idea of variability and how this diversity of life on Earth occurred. Diversity starts in this process of meiosis that we're going to go through today. Now, it has a lot of the same names of the steps that we went through last time, a lot of the same things that are happening. Cells or chromosomes are going to condense, spindle fibers are going to form, asters, all those things that we talked about in mitosis. Those are going to happen too. But there are some additional things that are going to happen as well, and that's all going to do with the variability. First of all, since I've been talking about variability, let's go back and talk about non-variability. Here they are. Remember that there are organisms that can reproduce asexually, not just our buddy the prokaryote, but that hydra on the left. You've probably seen a hydra under a microscope. Do you know who hydras are related to? What other animal? It's an animal. Hydras are an animal. Well, what other animal are they related to? One that we're real familiar with here in Florida. Every time I say that, people say dead opossums. I'm like, just because you see them on the road. Well, if I told you, and they're microscopic, if I told you these little things up here had stinging cells all over them, would that help? What if I told, come on, pencil. If these were all stinging cells that were sticking off these little projections. Do you know something else here in Florida that has these stinging cells that if you get too close to, especially when you're in water? Hydra lives in fresh water. These other ones that I'm talking about, most of them actually live in salt water. You've been down to the Keys, right? Right? Yeah, coral. Yeah, that's exactly. Hydra are freshwater species, what we call cnidarians. Corals fit into that group as well. Cnidarians are jellyfish. Anything that has these stinging cells is known as a cnidarian. The C is silent. But so this is an animal. It's not a prokaryote, right? It's a eukaryotic. This is a multicellular critter. And when it reproduces, what it does, one of the ways it can do it is by simply making a bud. Right? That's exactly what that is on the left-hand side. It's not its name. That isn't Bud. The next one wouldn't be, Chuck or something like that. But that's exactly what that is. This is really nothing more that at a much more advanced level than the prokaryote split in half. See, like these guys over here? Those are prokaryotic cells split in half. That's a neat picture, isn't it? The red part is the chromosome, by the way. So this is asexual reproduction. We get clones. This is asexual reproduction. We get clones. So what we're going to talk about is how things like you and I and redwoods and all these other species out there actually produce not asexually by just splitting in half, but through sexual reproduction. And again, sexual reproduction has a very defined definition. It's a very set definition. Sexual reproduction is the mixing of gametes, sperm and egg. It's that's what when organisms reproduce by one cell from one organism uniting with another cell from another organism, a sperm and an egg, when those unite, that's called sexual reproduction. However you do it, externally, internally, however you go about it, whatever act you engage in. For example, many of these corals, when they reproduce, the males, the sperm, they just let them out into the water. And then the females are in the water, they let all their eggs out in the water. Of course, the water is a great mixing place for sperm and egg. All mixed together, maybe fertilized, but it also means a lot of your sperm and egg are going to be eaten, aren't they, right? Because they also make good food. All right, how do we do it? Well, here we are down at the bottom. Let's start in the lower left-hand side and introduce you to a couple of terms. Multicellular is not one of those, but the word diploid, all right? The diploid number of chromosomes in a cell, regardless of the species we're talking about, can be mathematically represented by a quantity, a number, 2n. Whoops, 2n, there we go. You see it's 2n right there, right, 2n. Now, the number 46, I don't automatically assume 2n is always 46. In humans, that's what that picture is right there. It says that our 2n, our diploid number of chromosomes in our cells, is 46. Now, this is a good time to actually talk about, let's refer to organisms, or human beings specifically in this case, of having 23 pairs of chromosomes. It's better to really maybe think about it that way because you have a set of chromosomes that you got from your dad, right? If you follow the little cycle here, from the testes, meiosis occurred, which is what we're going to talk about, and sperm were produced. right? And then you also got a set of chromosomes from your mom in her ovary through meiosis. Eggs were produced. But now these sperm and these eggs, these gametes, are not diploid. Do the math. They're haploid. So the process of meiosis, in addition to all that variability I talked about, was maintaining the species chromosome number. Our chromosome number is 46. Now, there are cases where people have one or two or too many or too few, and those are problematic. But in multicellular diploid, biologically normal adults, we have 46 total chromosomes, better called maybe, or accurately called, it's 23 pairs. A pair from your dad, those are the paternal, and a pair from your mom, those are the maternal chromosomes. All right? So in this process of meiosis, as we go into the green part, or the blue-green part, whatever there, we're going to divide the number of chromosomes in half. So we're going to take a diploid cell, divide it in half, and we're going to get a sperm which has 23 chromosomes. In the case of humans, it would be different if it's were a watermelon, this would be 500 and 500. Remember, I said they have tons of chromosomes in watermelon for some odd reason. And 23 chromosomes are going to be found in the egg. Now, when they don't have the full set, as I said, then now they're haploid. Diploid, Di means two. Haploid means half a two, or one. And then when fertilization occurs, again, this is what I was talking about earlier, however you do that, however you go through this process, however you unite those gametes together, whether it's internally, externally, in the ocean, when the fertilization occurs, the number is restored. The 23 chromosomes in the sperm, if we're talking about a human, or the N, so we're going to use N to refer to any organism out there. So the N sperm gamete plus the N egg gamete make a zygote, a 2N. That very first cell, after the sperm and the egg come together, when that fertilization process, when that fertilization, which is sexual reproduction, and when that comes together, you make a diploid zygote. And then the zygote gets bigger by mitosis. Mitosis, quantity, right? So what we're doing in meiosis is we're making every sperm and every egg is a little bit different. We want that because then we want, when sperm and egg to come together, to have compound upon that difference. Think about that. Sperm are different. And by the way, not every sperm a male makes is the same. Not every egg a female ovulates is the same. They're genetically all different. So there's that genetic variability there. And then the sperm and the egg randomly unite with one another. So that variability on top of that variability on top of that variability means we all don't look alike. Right? We all don't look alike. We're all different. Whether we're talking about us, redwoods, wildebeests, or flatworms, it doesn't matter. We're all different through this process of meiosis, okay? Now, I do want to show you very quickly, this is not a botany nor a bio 2 class, so I'm not going to spend a lot of time on this, but I do want to show you there is alternative ways of doing this. For example, these are ferns in this picture that you see right here. I'll get you oriented. There's the zygote, right? The zygote grows up through mitosis, and it makes a sporophyte. The frond of the fern, it's not really a leaf for botanical technical reasons. We don't call it a leaf. We call it a, why am I stuck on caps? There we are. We call it a frond. But the fern frond is something we call a sporophyte. And if you've ever looked on the back of many fern leaves, and not all of them do this, but many of the more common ones, especially like all the resurrection ferns that I see all over the trees now, they're little spore-producing structures. And they produce spores through meiosis. Meiosis. So what do we know about the makeup of those spores if they've been produced through meiosis? What do we know about the differences or the similarities between those spores if they were made through meiosis? Remember what I said, meiosis makes cells, mitosis makes lots of cells. Meiosis makes what kind of cells? And in the spores are the cells, by the way. or the spores are the cells, excuse me. There you go, variable, right? And if one of them happens to survive, it germinates into something that we now call a gametophyte. This is the way plants, some fungi, reproduce through a process called alternation of generation. I didn't mean to write right through the word, but what we mean by that is in alternation of generation, there are two multicellular stages. The sporophyte is multicellular. There's mitosis, right? So the fern frond is multicellular, and the gametophyte is multicellular. Right? This is the gametophyte of a fern. This is a multicellular structure. By the way, all of those cells in that multicellular gametophyte, this is the gametophyte part. All of the cells there are haploid. So you don't have to go with the way we do it. You don't have to reproduce this way. These organisms still generate tons of variability, and they still survive, obviously. We see ferns around everywhere, but they just do a little bit different. They use these gametophytes as gamete-producing factories, basically, with the idea that most of your gametes aren't going to survive. Remember, most gametes aren't going to survive, so you make a lot of them, just like the reason why the male and the female coral just kind of spawn out in the water, all those tons of sperm. An egg. Now, don't worry so much about the alternation of generation. Just more so, just kind of understand there are different ways that organisms reproduce. But I'm not going to focus on that because that's something we talk about a lot more in bio too. We're going to go back to the typical way that we saw in the previous slide. This is what's called a karyotype. These are really cool. I used to have to do these in college, but they were in black and white. These are all chromosomes, and they've been dyed, if you will, different sections of the chromosome, different genes, different areas of the chromosomes. And then a karyotype is they go through and they match them all up. So you notice we don't have the numbers, these are human, 1 through 46. We have 1 through 22 pairs, right? Like the number 1 that I boxed, there's the number 1 paternal and the number 1 maternal. There's a term for that. These are terms that I'm going to be using later on, so it's important that we introduce them. The number 1 paternal and the number 1 maternal, the number 13 paternal and maternal, that those pairs are known as homologous. I'm typing it out. Homologous chromosomes. Homologous chromosomes are the copy, or not the copy, shouldn't say it, nope, not the copy. It is the maternal end of the paternal chromosome. So if they're the number 12 paternal, its homologous chromosome would be the number 12 maternal. You get the idea? So we're looking here, I'll get to the XY in just a minute, but at 22 pairs of homologous chromosomes. Remember, homo means same. Logos refers basically to structure. Logos refers to areas on the chromosome, so the same areas on the chromosome. Even though the genes might be different. If you remember, if you did any of that genetic stuff about dominant recessive, that plays out in all of this. But even though, not the genes will be the same, but the information on those genes might be different. All right? That cool picture. Like I said, when I was in college, we used to have to do these, but they were black and white. We used to have to match up all these banding patterns. That's how they would find all the homologous chromosomes. And they don't always lay out real nice and straight like this for you either. In human beings, the first 22 pairs of chromosomes we call autosomes. The last pair in any species, right? So that number 1 through 22 can change depending on the species. But the last pair is called the sex chromosomes. And you can see in this karyotype that the sex chromosomes are X and a Y down there in the lower right. So is this going to be a boy or a girl? Or is this from a boy or a girl, male or female? You got it. All right. Let's look at the simple picture of this whole idea of these homologous chromosomes coming together. So here we got a cell. First of all, let's go through it and kind of make sure that we orient ourselves as to what we're looking at. How many total chromosomes do you see in here? Chromosomes. Now remember, the way they're drawn, they've been duplicated. So I'm not asking about chromatids. I want to know chromosomes. Okay, How many chromatids do you see there? How many paternal chromosome, how many paternal chromatids do you see? Beautiful. Excellent. Excellent. All right. We're going to start moving through the stages of meiosis here in just a minute, but this stage that you're looking at right here is actually prophase 1. I'll come back through prophase 1 again in a second. This is actually a good time to mention the names. They're the same names. There's going to be an interphase and a prophase and a metaphase, and they're all going to look very similar. But don't confuse them when you're reading about them or on the exam. If there's a number that follows the phase, if I say metaphase 1 or anaphase 2, then I'm talking about meiosis. If there's no number, then it's mitosis. So that's a good thing to always remember. So when you're looking through the questions on the exam, you don't confuse those two together. So here we are in prophase, right? The chromosomes have condensed. They've duplicated. They're held together by those cohesion proteins. There's the centromere. All of that stuff. There's sister chromatids of one duplicated chromosome. Then down below, notice those are non-sister chromatids of a homologous pair. So in the lower left, the blue and the red, these down here, this might be the number 7 maternal and the number 7 paternal. But the chromatids are non-sister chromatids, whereas the sister chromatids are one duplicated chromosome. So this gets very confusing here pretty quick. But this is what's going to happen during prophase 1. We're going to say that these homologous chromosomes, right, are going to duplicate and make copies of themselves. Now, I'm going to step back for a second to talk about interphase and just quickly go through that because there's not much going on. And then we're going to get in more detail looking at prophase. I want to look at not only interphase, but I also want to look at the whole process of meiosis from start to finish, kind of like looking at the forest, and then we'll step back and look at the trees. All right, there's an interphase. Now, during the interphase of these cells, now, not all your cells in your body undergo meiosis. The only cells that undergo meiosis in your body are what we call your gonial cells, which are in your gonads. These cells have the ability to undergo meiosis. Your heart cells, your skin cells, your liver cells are going to undergo mitosis. That's all. They're not going to do meiosis. You're not going to be making sperm and egg out of your liver cells. All right, so during interphase of these cells, we have a G1S and we have a G2. Same thing. Same thing that was going on during mitosis and G1. They were growing and differentiating. In G2, the chromosomes were synthesizing and duplicating. In G3, they were getting ready to divide, and so on and so forth. So as we get to the end of interphase, so we look at that picture right there. Looking at that cell right there. Got a very simple cell here. It's got 2 chromosomes, right? It's got a blue one and a red one. Let's stick with the blue one being the paternal and the red being the maternal, just to keep them straight. So the paternal chromosome is going to make a copy. The maternal chromosome is going to make a copy. So this cell, when it's not dividing, it's got 2 chromosomes. But during this process of division, just like it did in mitosis, it duplicates all those. All right. Very good. Now here's where it becomes different. During meiosis 1, in which there's going to be a prophase 1 and a metaphase 1, an anaphase 1, and a telophase 1, during meiosis 1, this whole idea of going diploid to haploid plays out. Right? Meiosis 1 is sometimes called reduction division. Reduction division, what occurs, and I'll show you exactly how this happens, but overall what happens in reduction division is the number of chromosomes in the original cells divided in half. Now, take a look at that and make sure you understand what I just said there. So I'm starting with this cell up here. All right, how many chromosomes in that cell? Just to review. How many chromosomes in the cell I'm pointing at? How many chromosomes in each of these cells? Gotcha. And just to round us all out, how many chromatids in that cell? This is what it's like to draw a circle with a right-handed person when you're left-handed. How many chromatids there? Beautiful. All right. Now, Then after meiosis 1, telophase 1, there is no interphase 2. That's why interphase doesn't have a number. There's just an interphase. Cells don't stop and do any interphase stuff until they've gone through meiosis 2. So they run straight from telophase 1 right into prophase 2. Now, during meiosis 2, we call it mitotic division. And if you just follow one of those cells, like the one on the left, that looks like exactly what we saw in mitosis, right? The homologous, the chromosomes, the sister chromatids are held together, right? And then they pull apart, those spindle fibers pull them apart when the cohesion proteins disintegrate, and boom, we end up with one chromosome in each cell. All right, you told me there was one chromosome in each cell after meiosis 1. How many chromosomes in each of those 4 cells that you see after meiosis 2? How many chromosomes? How many does each cell have? Chromosomes. Now, chromatids, you're right, there are no chromatids. But these are now, you're only a chromatid when you're attached to something, right? Sister chromatids separate, becoming chromosomes. So each one has one chromosome, doesn't it? Each one has one chromosome, no chromatids. Chromatids are gone. They're only chromatids when they're attached. Think of chromatids again as being kind of state of being. Me and my copy are attached, we're a pair of chromatids. But together, we're a chromosome. So again, like I said, reduction division. We divided the number of chromosomes. We went from diploid to haploid after step 1. And then in meiosis 2, they call it mitotic division because it looks a lot like mitosis. Matter of fact, once we get through meiosis 1, meiosis 2 will be very simple. We can fly through that because we talked about that already. All right, so let's back up for a second. Let's go up to, where does my little chart start? Prophase 1. There we go. All right, so this is meiosis 1, meiosis 2. This is all the steps together. But what I've done is this is kind of showing the overall thing again. I got a couple things I want to talk about here. And then in the next few slides, I've separated each of these steps. So we're going to take this slide and break it down a little bit further. All right, after meiosis 1, here's what it does. 2 end to end. Meiosis 1 is reduction division. That's the purpose. By the end of meiosis 1, by the time we get right here, once those cells completely divide, and by the way, they're going to divide cytokinesis-wise the same way if they're a plant and an animal. I'm sorry, the same way they would if they were a plant and an animal. Animals by cleavage furrowing, and then plants by cell plate formation, the same way. So the cytokinesis is going to be different, or it's going to stay the same based on what they are, but it's different in meiosis depending on whether you're a plant or an animal. But the purpose of meiosis 1 is divide the number of chromosomes in half, diploid to haploid. The purpose of meiosis 2, we talked about it being mitotic division, but it's a little bit different. It's set up a little different based on some things that happen. At the end of meiosis 2, we're going to make 4 haploid daughter cells, okay? 1 gonial cell, that's this guy right here at the beginning, prophase 1. After it divides twice, divides here, is going to end up with 4 haploid daughter cells that are, and this is important, that's why I put it in red there, genetically non-identical. They're different. They're variable. They're not the same. That's very important, remember, because I'm saying what we're trying to make here is variable cells, variable sperm, variable egg. Nobody wants to be the same. You can't. Organisms can't survive that way. All right, now, what's going on in prophase 1, we'll talk about it as we get a little bit of detail, but you do see in this picture right here that the chromosomes have not only begun to duplicate, but the homologous chromosomes are finding each other, right? So during prophase 1, let me jump up a couple slides. That'll be fine. All right. Oh, sorry. Not that far. All right, that's what I mean. During prophase 1, if you look down here, so I'm looking at this cell right here. Let's kind of orient ourselves. All right, we got the blue chromosome, and it's got a copy. We got the red chromosome, and it's got a copy. It may not look real obvious in the picture, but what you're seeing, like, for example, right there, would be a pair of homologous chromosomes. The number one paternal, the number one maternal. Each of those would have their chromatid. So there's actually 4 chromatids across there. As a matter of fact, we say that when these homologous chromosomes seek each other out, they don't call each other out, it's chemical cues. They lay next to one another, like you see in that picture. When they're laying next to one another, we say that they are in something called synapsis. Syn, S-Y-N, is side by side. So the number 5 paternal chemically recognizes the number 5 maternal, and they get up next to one another. And the number 1 paternal and maternal. Now, does that always happen exactly? No. Doesn't always happen exactly right. Most of the time it does, thank goodness. But there are times in which deformities occur because the wrong homologous chromosome lined up, like a 3, number 3 paternal, lined up with the number 8 maternal or something like that. They're like puzzle pieces, though, chemically, so that's how they usually get it right. Okay, when they're all laying in synapsis, I said you got 4 chromatids there. So under a microscope, we sometimes, when we look at these things, they look like tetrads. If you look at the one next to it, in metaphase 1, you can see the tetrad. You see the 4 chromatids much more easily. Okay, so before I leave prophase 1, right, let me review once again what's happened. The homologous chromosomes have seeked each other out, right? They found each other. They're in synapsis, right? And they form these tetrads. Now, other things that have happened. The nuclear envelope is disappearing. The spindle fibers are forming. If you're an animal cell, the centrosomes and the asters and the centriole pairs, they're doing their thing. If you want to kind of just put in your notes all that other stuff, see mitosis, that's fine. That's what's going on. All that other stuff that's going on with the nuclear envelope and the spindle fibers, all that's going on. The only difference is what the chromosomes are doing. They didn't seek each other out during mitosis. They just made a copy and split in half. These homologous chromosomes are seeking each other out. Now, I'm going to go to a whiteboard for a second because I need to draw something for you. Let's talk about why they're doing this laying next to one another. And this is probably one of the, well, this is the most important thing that happens in prophase 1, especially when it comes to variability. So I'm going to draw some chromatids. I have to do this with the pencil, though. There's no way I can do it with the mouse. So let me get my pen to start working here, folks. Well, I'll be fine. All right. I'm going to draw some chromatids. I'm going to draw some homologous chromosomes in synapsis. Now, yes, I know they kind of look like flatworms, but just stick with me. And I'm going to draw the pair of homologous chromosomes, like I said, in synapsis. Now, the chromatids are connected together, but the homologous chromosomes, they're not connected together. They are kind of connected together, but hold on a minute, I'll explain what I mean by that. Let me get some different colors here. Now, what I'm going to do is I'm going to pick some genetic traits, some human genetic traits that only have 2 choices to make this simple. Like, for example, well, eye color is not a great one because of the fact that we find that it's not as simple as that. Earlobe attachment, right? So here's your head. If your earlobes have a little bit of a droop to it, we call them unattached. And if your earlobes, that's not really a very good head, but if your earlobes kind of go straight in, or there's no lobe, then it's called attached. All right. So we use that one. Let's say will indicate that genetic information is being found on this part of the chromosome. I'm going to use the tips only to make it easier for me to draw, okay? So what we're saying here is that if we looked at a chromosome and we wanted to find out where is the location for the gene for what your earlobes look like, we would be looking on that chromosome for something called a locus, not the insect, but a locus, a location. So what I've drawn is I've said, okay, right up here at the top is the location for the gene for earlobe attachment. It could have been anywhere along this chromosome. Like I said, the only reason I've done it up here is to make it simple. Well, this one next to it is a copy of it, so obviously the information is exactly in the same place. All right, now we're going to go on the other side of the chromosome to the other homologous chromosome. And since they're homologous, they have the same genes, right? But they might have different information about those genes. So let's say if we leave it blank like this, we're saying that so we'll say this is unattached earlobes and this is attached earlobes. Scratch that. There. Make sense? I'm sorry. I know that I draw for crap. Excuse me. All right, and we need another trait. Whether you can roll your tongue. There you go. You can either roll your tongue or you can't. That's another simple one. And we'll say the locus for that, I need a different color. The locus for being able to roll your tongue is down here on the bottom. Now, genes aren't always at the bottom. Again, I'm just doing this for sake of simplicity's sake. And when it's colored in, we'll say that you can roll your tongue. Don't I color well, especially my right hand? Homologous chromosome is in the same spot, but we'll leave it blank and we'll say that you can't roll your tongue. All right? So over on the left-hand side, we've got the genetic information that says you can't roll your tongue and you have attached earlobes. And on the right-hand side, you've got unattached earlobes and you can roll your tongue. Right? Does that make sense? Yeah, I apologize for my lack of drawing ability, but does that make sense what I'm setting up? And I'm only looking at 2 traits. Good. All right. Now, another thing that's going to happen during prophase 1, and as I said, this is extremely important for variability, so you're going to have to really bear with me on this because I'm going to have to. If I hit that line, let me see if I can erase just part. That doesn't work very well at all. All right. The next thing that happens in prophase 1, I'm sorry, folks, I'm having troubles with my art here. The next thing that happens is something called crossing over. Really important, really important. Here's what happens in crossing over. When those tetrads are laying side by side, randomly, every time they do it, randomly, folks, variability, randomly, those non-sister chromatids are going to cross over one another. And they do it randomly up and down this set of tetrads, half dozen or so times, or maybe even less than that. They don't have to do it a whole lot of times. But just for the sake of the argument, I'm going to say that what happened is these 2 inside chromatids crossed over one another. So if you'll let me kind of extend the drawing down here a little bit, it might help. So I'm going to talk about these 2, this one here and this one here. So the one on the right curved over, and the one on the left curved over. They actually cross over. It's actually called a chiasma. That's a crossing over point. Chiasma. Remember, CH is ka in science, not. I always remember it's chiasma or chiasmata. Now, chiasma 1, chiasmata plural. So there may be several or a handful of chiasmata up and down there. But that crossing over point. Now, go back to my colors again so we keep track of that. It was these 2 that crossed over. So the one underneath is going to be the one on the left, and the one on the right is going to be the one right here. All right? So, back or forward. Does that make sense? That's kind of the setup. Let me go back to the slides now. Right there. All right, so see on the left-hand side, meiosis 1. So now there's 2 chiasmata here, right? And they did it at the tips as well. But you get the idea, they're going to cross over, okay? And I'll talk in just a second about what this is all about, but I want to make sure you understand the structure. And importantly, though, I want you to understand the structure of the spindle fibers. If you look at metaphase 1, and you compare that with metaphase of mitosis, you got to be really careful when you're studying this stuff, folks, because the names sound the same and the processes sound the same. It's very easy to get them confused. That's why I spend a lot of time kind of comparing and contrasting them. If you take a look at the pair of homologous chromosomes in metaphase 1, there are spindle fibers connected to the kinetochores of the outside chromatids, but not the inside. Right? There are no spindle fibers connected to these guys here. The cohesion proteins are holding them together, yes, but the spindle fibers are only on the outside. And if you remember, the spindle fibers are what pull these things apart. And just like in mitosis, when the spindle fibers form, they're under tension. They're pulling. Now, it was before, it was the protein, the cohesion protein that was keeping them from coming apart, but now it's the chiasmata. The crossing over points act like a weave, and they hold those tetrads together. All right, not for very long. I want you to make sure that you understand how that's different in metaphase 1 of meiosis, which is the next stage I'll show you in a second. It looks just like metaphase, versus metaphase and mitosis. Notice in metaphase and mitosis, each chromatid, there are only 2 there, each one has spindle fibers. All right, and again, understand the spindle fibers are the things that make the chromatids move. So meiosis or metaphase 1 looks just like metaphase in mitosis, except the chromosomes are all paired up. The homologous pairs are paired up. All right, now, anaphase 1. During anaphase of mitosis, these spindle fibers pulled that chromatid that way, it became a chromosome. These spindle fibers pulled that chromatid that way, it became a chromosome. And this happened because an enzyme ate away that protein. When you're in meiosis in metaphase 1, it's not the cohesion proteins, but these spindle fibers are going to pull this pair of chromatids, right? And these spindle fibers are going to pull this pair. Now, I said the chiasmata are holding them in place. Eventually, the chiasmata give and they break. Wherever they were crossed over, listen carefully to what I'm saying too. Like in this place, they're crossing over at the bottom, crossing over at the top. Wherever they cross over, they swap genetic information. So back to my picture where I was using the earlobe attachment, and I was using the rolling of the tongue. On the gene that had the earlobe unattached and the tongue rolling, and now swap that tongue rolling information to the other chromatid. We're swapping stuff up, right? We're generating variability. So then, whoops. If you look at the anaphase 1 picture, you can see that on the blue chromosomes there's a little bit of red, and on the red there's a little bit of blue. Now that's actual molecular material that's been swapped. It's what we call recombinant DNA. Recombinant DNA is DNA that's been put together in a different way. It's been taken apart and put together in a different way. And it's been put together in a way that's variable, because where those chiasmata form each time is variable. It changes. They don't form along the same tetrads in the same place, in the same cell every time. And there's millions and trillions of, I think the odds are something like 16 trillion, 1 in 16 trillion of having an identical offspring that wasn't an identical twin. Like you had a baby now and 3 years later, same parents had the same identical baby. It's like 1 in 16 trillion are the odds of something like that happening. Some crazy number. All right, so then we're going to move from anaphase 1 into telophase 1. And in telophase 1, cytokinesis is going to begin to occur, different on plant and the animals. And you can see that the nuclear envelope is going to begin to form. Now, we're going to turn around in the next step of prophase 2 and get rid of it, but got to keep those chromosomes protected even for this little bit of time. You notice, too, that these are obviously animal cells because we have centrosomes and centrioles, but you also notice that the spindle fibers are now gone. So these are things we talked about in mitosis as well. Okay, so let's do the numbers game again. Let's imagine that we are at the end of cytokinesis. We now have 2 cells. How many chromosomes in each of those cells? Chromosomes. How many chromatids in each one of those cells? Excellent. Thank you, Ryan. All right. And we started up here at the top with 1, 2, 3, 4, 5, 6 chromosomes, right? So we've done the reduction division part. So we're going to jump now from telophase 1, cytokinesis, we're going to split those 2 cells. So now in the next set of sequences, we're going to be following the fate of 2 cells, all right? Right there. So those are the 2 cells. So we're in prophase 2. Now prophase 2 is nice compared to the other prophase stages because there's really just sort of a holding pattern. The nuclear envelope's going to disappear. If it's an animal cell, the centrosomes and the central pairs are going to do their thing, and the spindle fibers, like you see in the picture, are going to form. All right, but also notice what's different than mitosis is if you look at those chromosomes, they're mixed up. There's some red in with the blue and some blue in with the red. All right, now I remember at the beginning I said meiosis 2 is sometimes called mitotic division. And if you look at what's going to happen in the next few stages, it looks almost exactly like mitosis. If those chromosomes were all the same color, like we did in the last chapter, it would be no different, would there? So in metaphase 2, those chromosomes are going to line up on the metaphase plate. And now the chromosomes look like they did when they were in mitosis. Even though the colors are all mixed up, now the chromosomes are arranged like this. So this would be metaphase 2 if this were meiosis. This is mitosis, but if this were meiosis, that'd be metaphase 2. So there's kinetochore microtubules on each one of those kinetochores. So when they pull, now guess what happens? Now the chromatids are going to separate. They're all different and mixed up, but they're all going to separate. They're going to move to the opposite ends, and then telophase 2 is going to follow. Right? Nuclear envelope is going to form, and we're done at that particular point. Cytokinesis is going to happen, and we're going to end up with what we call 4 haploid daughter cells. More importantly, though, is 4 haploid genetically non-identical. They're not identical to one another. They're different. Each one of those daughter cells is going to become a sperm or an egg. So we got variable sperm and variable egg, because remember I said the crossing over point, where they come through the chiasma, that's random each time. Even in metaphase 2, look at that. See the way they're lined up on the plate? Left to right, they don't always line up the same way. Like, for example, in the top cell, the right chromatid is to the right in the top one. The red one with the little blue is on the right in the middle one, and then the all blue one is on the bottom. Well, left to right, that could be the other way around. It's all just randomly how that comes together. Now, this is a great slide because it does a good job of helping you keep straight the difference between the 2 stages, or the 2 events, right? Mitosis and meiosis. The next one is actually the text of all of this. I'm not going to read through all of this, but this is a good thing to obviously to go through and make sure you understand, and you can describe what's happening in these stages. So this is a visual of that, right? So the parent cell has 6 chromosomes. During mitosis, chromosomes duplicate. That's it. They don't they don't engage in synapsis. They don't make a tetrad. They don't cross over. They then line up on the metaphase plate, and they split. Boom, end of story. We're done. Daughter cells through mitosis have the same number of chromosomes. You had 6 chromosomes. You duplicated to 12 chromatids. You then divided those chromatids and gave each cell 6 chromosomes. In meiosis during prophase, we're going to cross over, right? We're going to form the chiasma. So in metaphase 1 of meiosis, we're going to have this metaphase, but it's going to be the pair of homologous chromosomes that line up. And again, remember, the way they line up is not necessarily, like in this case, the blue on the left and the red on the right, and then the red and the blues on the right. They can be different each time. Different possibilities. I think for each human, I said 16 trillion, more like 8 billion different ways this can all happen. These crossing over points and where they line up on the metaphase plate. Because think about it, where they line up in metaphase 1 is going to determine which cell you go to. So the 2 blue ones in the second and the third are going to go to the cell on the right because they're on that side of the cell. And the red ones, the 2 red ones, the second and third, are going to go to the cell on the left. And then those are going to divide, right? Those are going to split. Those chromatids are going to split, and we're going to get 4 genetically non-identical daughter cells, each with half the number of chromosomes, and each, look at the picture, different. The chromosomes are different. The coloring, again, representing the different traits I was talking about. So we have 4 genetically identical daughter cells. Now that's going to happen again whether we're talking about mitosis or meiosis, either one. I'm sorry, I didn't mean to say mitosis. I meant to say just in meiosis, not in mitosis and meiosis, excuse me. Okay, makes sense? Any questions on that? I just got a few more slides where I get back to this variability thing that I really like to talk about. See, remember, mitosis is just making identical cells. Its job is quantity. I mean, do variations occur? Well, yeah, because like I said, every time you duplicate a chromosome, you don't always get it right. But here we got the crossing over points that cause variability. Where they line up on metaphase 1 causes variability. Where they line up on metaphase 2 increases the variability. So if you know anything about probability, whenever you take the odds of something happening and then the odds of something else happening, you multiply those. So if the odd of something happening is 1 out of 4, and the odd of something else happening right after that is also 1 out of 4, then you got a 1 out of 16 chance, 4 times 4, of this happening. All right? So you can just imagine the possibilities when it comes to the ways we can arrange these chromosomes. And of course, then there's also the variability about which sperm and egg do the fertilizing, right? So let's take just a step again with this whole idea of variation. Humans create about 65,000 mutations a day throughout the world. Throughout the world, on average, we do this because we know that there's a kind of a clock at the way mutations occur. It's a molecular clock. About every X amount of DNA replications through X amount of humans, we get a mutation. So about 65,000 mutations a day. Now, with human beings, most of those mutations are silent mutations. I talked to you in class before about silent mutations. There's part of your DNA in eukaryotes, not in prokaryotes, but in eukaryotes, that's known as your introns. And your introns are parts of your DNA, A's and G's, that aren't read, that aren't used, that aren't turned into proteins. They're there for reasons that have to do with, I think I mentioned before about this idea of selfish genes, genes just making copy. Okay, I mentioned to you that where they assort during metaphase 1, independent assortment, we get variability from that. So if you take a look at something like this, here's the same cell, same number of chromosomes, but I've arranged, they're not crossed over, just to make simplicity's sake, but you can see that there are a variety of ways of arranging these 6 chromosomes. Right, I could make all the blue on the left and the red on the right, or all the red on the left and the blue on the right, or some combination in between. If you want to know how many possible ways this can happen, what you do is you take the number 2 and you raise to the exponent the number of pairs of chromosomes. We have 23. So human beings can produce over 8 million different types of gametes. 8 million. 8 million. And remember, what I was right about the 16 trillion. So if it's 8 million for the male and 8 million for the female, 8 million times 8 million is, I think, 16 trillion, isn't it? It's a big number, whatever it is. So that's the possibility of 2 parents having a child and then, having one 3 or 4 more years later and having that child be identically genetic. It means that when those parents made their sperm and egg, they chiasmed in the same place. I just made a word out of chiasma, but they crossed over in the same place. They lined up on the metaphase plate the same way. The same sperm fertilized the same egg. Everything happened. Is that possible? Yes. 1 in 16 trillion times. How many people are there on the planet? Certainly 16 trillion, right? It's 8 billion, 7 billion, 8 billion, right? So more possible ways of making humans than there are. Now, have there been 16 trillion humans alive since the beginning of time? That's a good question. I'm not sure. Bet you can find that online somewhere. Yeah, it's a big number, whatever it is. All right, so just again, another little review. I just want to make sure we got the pairs of the chromatids, synapsis and crossing over occurs. They make there's a really cool picture of a TEM is transmission electron microscope. There are 2 types. Well, there are probably more than 2, but there's also a scanning electron microscope. You can see the crossing over points. Again, you can't really make out the chromatids, but there are 4 chromatids there. And then, of course, then they break apart, and they break apart again. There's that word recombinant that I used before. Recombinant chromosomes, in this particular case, the way the artist drew it, 2 of the cells, the one on the far left and the far right, the chromosomes are not recombinant. It's all the red or all the blue. But the ones that cross over, when they switch their genetic information, they recombine their genetic information. So they're called recombinant. There it is. Humans average about 1 to 3 chiasmata per chromosome pair. So every time we go through meiosis, all 23 pairs of your chromosomes undergo about 1 to 3 different crossing over points. Different, random crossing over points each time. All about variability. And then, of course, the last thing that increases the variability is this idea of random fertilization. Oh, there's the numbers. Each with about 8.4 million possible chromosome combinations produces a zygote of about 70 trillion diploid combinations. So in other words, it is possible to have an identical offspring 1 out of 70 trillion, which is a heck of a lot bigger number than what I was saying. I think, what is it? Million, billion, gazillion? Trillion? Maybe it's just trillion. I can't remember. And then again, we don't know which sperm is going to fertilize which egg. And there's all these very, very interesting things we found out about sperm, believe it or not. There are sperm have different jobs. For example, the vagina is not a horribly hospitable place for sperm. It's somewhat acidic. And if you're thinking, well, why would that be? Well, the vagina is an opening from the outside to the inside of your body. And anytime there's an opening like that, the body tries to protect that opening. It doesn't want anything going in there. So the vagina is a little bit acidic. So the first sperm on the scene actually neutralize the acidity. They kind of, if you will, give their lives for the sperm behind them. And then those sperm behind them enter into the uterus, and there's 2 little tiny holes at the top of the uterus. One goes to one fallopian tube, and one goes to the other, and there typically is only an egg in one. So you got to make the right choice. And if you go in the wrong fallopian tube, you can't turn around and come back, because by then your buddies are already way up the other one, and somebody's already done the deed. So there's a lot of variability that occurs in that. There are even some rambosperm in some species that actually hang behind. They don't try to go up the fallopian tube. I call them rambosperm because what they do is they actually assault and destroy sperm from other males. So if a female were to mate with a male and then soon afterwards mate with another male, the first male's rambosperm would try to kill them because it wants its, somebody related to it to do the fertilizing. I read a book about it. It was amazing. I was like, holy cow, all this stuff that's going on among sperm. Who would have known? All right, now this picture is not in your book. It's not in this chapter. I have to look if it's in one of the later chapters, but it's a relatively simple picture. I want to talk about how sperm and eggs are created. Now, we already talked about it, but I want to talk about something specifically that happens in this idea of gametogenesis, the production of gametes. Let's do the simple one first. So there's a gonial cell, a gonad cell. What's that number? 107 billion, right? Billion, yeah, 107 billion. Holy cow. And what we said, 70 trillion. So we haven't even exercised all the possible ways that humans could look. I guess that's good. You won't get bored, right? Thanks for that. 107 billion. Good God. All right, so you got this gonial cell, and a gonial cell is diploid. Now, gonial cells can undergo mitosis too. You can make more than one gonial cell, but when they undergo mitosis, they're going to make identical gonial cells. It's only when they undergo meiosis, like you see here, that you get the variability. All right, so a primary spermatocyte, the gonial cell you'd find in a male, undergoes meiosis 1 and goes diploid to haploid, 2 secondary spermatocytes. All right, each of those then divides in meiosis 2, and we get 4 spermatids. You slap a tail, an energy pack, that's kind of what that is, and you let that sperm go. The flagella goes off and tries to find itself an egg somewhere, right, where we get the fertilization. Now, a little different in females. And this is kind of interesting to me. I read a book a number of years ago about maternal care of their offspring. About, different species, it's both the male and the female involved. Some species it's the male. You might have heard about seahorses, right? In some species, or many species, it's the female. And in many species of mammals, it's just the female. So female mammals undergo a type of gametogenesis known as oogenesis. They're producing an ovum or an egg. It's ovum singular, ova plural. Those are eggs. All right, so we start with a primary oocyte. That's the gonial cell. And again, it can undergo mitosis and make other primary oocytes, but they're the same. But here's where the difference becomes. During the process of meiosis II, you're only going to make one secondary oocyte. Okay, that other secondary oocyte becomes something called a polar body, and it actually remains attached to the cell. Now, females stop meiosis at that point when they're born. In other words, females are born with all the secondary oocytes they're going to have. So their primary oocyte has undergone meiosis I during fetal development. So when that young girl is born, all of her secondary oocytes have already been made. They're all made. Remember, females don't reproduce all their lives. They start, during puberty, they end in senescence or menopause, so X number of years. So all those eggs have already been made, well, not made, but all those secondary oocytes have already been made, okay? Now, you begin as a female to ovulate one of those secondary oocytes. Of all the ones that have been made, they're kind of, actually, if you look, it's very interesting in the ovary. It's like a conveyor belt. Eggs are moving around, and then all of a sudden an egg is ovulated. About every 28 days in a human, starting at puberty, an egg is ovulated. And that egg actually goes out into the space in the body right between the ovary and right between the opening of the fallopian tube. There's actually a small little space. The ovary, so like this is the ovary. The fallopian tube has little fingers to help catch the egg, and the egg actually goes out into the body cavity, finds one of those fingers, and enters in the fallopian tube. The reason why I mention that is sometimes the sperm gets out the fallopian tube before the egg gets in it, and you can get pregnancy in the body cavity. That's obviously not good. The womb is made for that. All right, so you ovulate those eggs. One a month, one every 28 days until senescence, and then you don't ovulate anymore. Now, if that egg is not fertilized, if that secondary oocyte is not fertilized, during a female's menstrual cycle, what she sheds is a secondary oocyte. Mother Nature is very conservative, okay? She's not going to bother to go through meiosis II with the secondary oocyte if it's not going to get fertilized, and if it's just going to get shed. Why waste the energy? Why waste the materials of the chromosomes? All of that if nothing's going to happen to this egg. So when a sperm fertilizes an egg, what it really does is it fertilizes a secondary oocyte. When the sperm fertilizes the secondary oocyte, the sperm waits for a while. The egg finishes meiosis II and makes another polar body, right? You get a polar body because that's where the other cell is. You get a polar body from meiosis I, and that's still on there. The sperm fertilizes the egg, not the polar body, although maybe there are times that happens accidentally too. Then the sperm fertilizing the egg tells the egg, okay, prophase II, metaphase II, meiosis II, it's going to divide again, make another polar body. The polar bodies disintegrate. Nothing happens to them. And what we have left behind is one haploid egg, and since now it's got the sperm, we call it a fertilized egg. And when the nuclei fuse of those two, right, when the nuclei fuse, then we get junior. Then we get a zygote. And that zygote is only one cell, and we need to make a lot more cells, and so we're going to undergo mitosis from that point. Of course, as you know, as we get old enough, and our bodies are going to undergo meiosis, even though females have already undergone parts of meiosis before they're even born. That is some of the problem with females when they get pregnant older in life, because males, we make sperm if they're not used, we just recycle. So we don't really have old sperm sitting around, if that's a way to describe it. But females, they make those secondary oocytes, they're born with them, and then throughout their lives, they are exposed to chemicals or things happen that could affect the genetics of those. And so some of those secondary oocytes could be damaged. And they might be damaged when, maybe a young woman lived a little too close to a nuclear power plant or something. And 20 years later, 10 years later, she decides to get pregnant. And, that particular oocyte was damaged, and that's the one that was ovulated. That's why we talk about females being older getting pregnant is more problematic, because the eggs are, for lack of a better way, they're older. Although I've read some research lately that suggests we used to more readily attribute Down syndrome to females. Down syndrome would be a problem in the egg, the secondary oocyte dividing again. And if it's an egg that's gone bad, if you will, and it's an older female, we many times believe that was the cause of the Down syndrome. And I'll show you what Down syndrome is in just a couple of chapters, but Down syndrome is chromatids that don't separate properly. That's a problem. Remember the end checkpoint of mitosis, making sure everybody's got a spindle fiber? Yeah, something gets screwed up there. But I read the other day that actually males, even though we produce sperm and we recycle them, that if those sperm at the bottom aren't used, they just get torn down and recycled. That as males get older, the whole process of spermatogenesis becomes problematic. Things don't always work the way they should. And now there's medical research and other research to show that males are also capable of contributing some degree of portion to the genetic deformities like Down syndrome and that sort of stuff. So you can have old eggs, right? But it seems that the old sperm too. So even though males can reproduce until they're dead, there seems to be a greater chance of your spermatogenesis process not being quite as efficient when you're Hugh Hefner's age versus a young 20-year-old. All right. Any questions on that, Ryan? That's the lecture for the day. I'm going to turn the recording off.